Okay, so I was asked to give a bit of an overview regarding imaging of the blood-brain barrier in aging and age-related neurodegenerative disease. Next slide. If you could just run through these, Dendrong. I'm going to be beginning by talking a little bit about the function of the blood-brain barrier, and then I'm going to be talking about the different measurements that have been employed to look at this. And finally, I'll describe application of imaging techniques to investigate changes that occur to the blood-brain barrier in aging and age-related neurodegenerative diseases. Next slide. So in 1909, Goldman and colleagues injected a blue pigment into the blood of a mouse, and they observed that this distributed pretty much everywhere in the body, a notable exception being the brain. They concluded, therefore, that the structure of the vessels that perfuse the brain must be distinct to those that serve the periphery, and a key function of these vessels is to serve as a barrier. And so a key function of the blood-brain barrier is to prevent against unwanted infiltration of potentially harmful substances from the blood into the brain. But of course, at the same time, the brain is a notoriously well-perfused organ. This is a vascular cast of the human brain. And it's been estimated that if you line up all the capillaries in the human brain, they stretch to 400 miles, giving a surface area of 20 square meters. And this huge surface area is to facilitate a constant interaction and communication between the blood and the brain to allow essential substances to exchange across the barrier. And it serves an essential role in the delivery of, for example, oxygen and nutrients from the blood into the brain, the clearance of byproducts of cellular metabolism, and potentially harmful substances such as amyloid. And for this reason, the blood-brain barrier is often referred to as not a barrier, but the blood-brain interface. Now, damage to the blood-brain barrier has been observed in many common age-related neurologic conditions. And there is increasing evidence that this damage could be a key upstream event that plays an important causal role to drive downstream neurodegenerative processes. And in order to fully understand this hypothesis, we need to develop sensitive and practical measurement techniques in order to fully realize the potential of measurement of blood-brain barrier integrity as a novel diagnostic and therapeutic target. Now, in terms of measurement techniques, there are, to my understanding, basically two main categories. The first, and this is far more commonly employed, is to harness an intravascular contrast agent and simply measure the rate at which this crosses the blood-brain barrier and accesses the extravascular brain tissue. The second approach, and this is less common, but there have been particular probes that have been designed that adhere to particular elements of the blood-brain barrier. So for an example of the second category, this is an iron oxide particle, a MR contrast agent that is conjugated to an antibody that then adheres to regions of inflammation, in this case, in the penetrating arterioles in a rat brain that's undergone a stroke. But in regards to the first category, much more commonly applied method, the extravascularization of contrast agents, the choice of contrast agent is really important in terms of which aspects of blood-brain barrier permeability you are looking at and how this may change in pathology. So for example, focusing on the young healthy brain, first of all, the first image shows a map of gadolinium egress into the brain, and you can see that very little gadolinium actually makes its way across the blood-brain barrier in the young healthy brain. Contrast this to measures of endogenous water, and you can see that the brain just lights up and water readily crosses the young healthy blood-brain barrier. And according to this particular technique, it takes about half a second to do so. So just to dwell on this a little bit more, next slide, please, Tendril. The choice of intravascular contrast agent is really important. And so, for example, in the case of gadolinium in the top right, it's considered that gadolinium isn't normally able to cross the intact healthy blood-brain barrier. And when this happens, this is thought to reflect, therefore, paracellular transport, reflecting a breakdown in the tight junctions between the cells. In the case of the middle part of the figure, there are particular probes that have been designed, particularly using nuclear medicine, that probe specific transporters at the blood-brain barrier. And in the case of endogenous water, this is known to readily cross the healthy blood-brain barrier, and it does so via a number of different routes, including via aquaporin 4 at the end feet of astrocytes. Just to give an example as to how this kind of manifests experimentally, there was a study by Timothy Duong's group where they showed increased permeability in a rat model of stroke to gadolinium, but they showed a reduction in water permeability at the blood-brain barrier. So moving on now to the different measurement techniques that have been employed to look at blood-brain barrier integrity and function, focusing first of all on non-imaging techniques. There have been many studies that have used a direct assessment in animal models, and this basically involves injecting a contrast agent into the systemic circulation, in this case, Evans blue, and then perfusing the brain to remove the blood, taking it out, chopping it up, and performing high resolution microscopy to examine the distribution and extravascularization of the contrast agent across the blood-brain barrier. There have been techniques which build on the classical Ketty-Schmidt methods, which enable the first in vivo measurement of blood flow in the human brain. And basically, if you can take very careful measures of the concentration of a contrast agent in the arterial and venous side, you can measure the extraction fraction, the proportion of that label that makes its way through the vascular bed and into the veins in a single pass. And doing this using a global PET measure of label water uptake, in the second part, you can see there's a beautiful correlation between CBF, modulated with different levels of CO2, and extraction fraction. And at basal CBF, about 90% of the radio-labeled water is extracted during a single pass through the vascular bed. The final technique that I wanted to mention, and this is something that's increasingly being applied clinically, is taking blood samples and then taking CSF samples via lumbar puncture, calculating the concentration of endogenous albumin, looking at the ratio as a non-invasive surrogate of blood-brain barrier integrity, since endogenous albumin is not thought to be able to readily cross the intact blood-brain barrier. Moving on now to imaging techniques, DCE MRI has been applied prolifically to map blood-brain barrier integrity to gadolinium. And the way this method works is you inject gadolinium into the vein, you then take serial T1 weighted images, and then you look at the time course of the data to try to disentangle the arterial input function from the kind of residue function that reflects the delivery of gadolinium across the blood-brain barrier. And doing this, you can generate beautiful images that reflect the egress of gadolinium across the blood-brain barrier. And in the example shown here, you can see clear differences between a patient with small vessel disease and a healthy control. And I stumbled upon a nice review article that was published last year that describes the methodological pitfalls when applying these methods, which I gather is quite challenging when looking at neurodegenerative diseases where the rates of infiltration are fairly subtle in comparison to applying this technique to look at brain tumors or stroke, for example. Moving on now to nuclear medicine techniques, PET and SPEC. This has, of course, the great advantage of increased specificity compared to MRI. And this has been exploited to design particular imaging probes that act as substrates for specific transporters at the blood-brain barrier. So in this example, this is a PET substrate for the P-glycoprotein, which is an essential efflux transporter for the removal of amyloid from the brain. And they've shown that when they give a drug here, this results in a blockage of these transporters, resulting in increased accumulation of the tracer within the tissue. There's also been PET ligands that have been designed that adhere to particular components of the neurovascular unit, in this case, aquaporin 4 at the blood-brain barrier. And they've showed a reduced uptake using this tracer in the aquaporin 4 null knockout mouse as expected. The next technique is quite tricky. So it's an MR method, and it involves injecting an MR contrast agent, most commonly gadolinium. But here, unlike DCE, we're not trying to measure the egress of this external contrast agent across the blood-brain barrier, but we're exploiting the fact that this gives a difference in relaxation times of the water in the blood versus that in the tissue, and then we're then using it to estimate the permeability of the blood-brain barrier to water. And there are a number of different approaches that use this technique. This is nicely summarized in this review article. But just to give an example here, in the rat brain, they've taken serial T1 maps at different concentrations of gadolinium at different time points and used this to estimate the permeability of the capillaries to water. The next kind of family of techniques I've categorized into completely non-invasive methods that do not require an external contrast agent. This uses MRI and most commonly harnesses the principles of ASL. And because it's ASL, we're now looking at the permeability of the blood-brain barrier to endogenous water. And the great advantage of these completely non-invasive techniques is they allow repeated measures to be a lot more practical. And this has been exploited to, for example, show good scan, rescan reproducibility of the diffusion-weighted method of looking at water permeability across the blood-brain barrier. And another study recently showed that you could perform a baseline scan, then give a drug to the patient inside the scanner, in this case caffeine, and record a fairly subtle but very reproducible decrease in extraction fraction following a caffeine challenge. The basic idea of this technique, as I say, it relies on ASL. This is a method, which I'm sure most of you are very familiar with, but just briefly, it involves applying a labeling pulse that intersects the neck, and water molecules in the feeding arteries on their way to the brain then become magnetically labeled. Next few animations, please, Dendron, thank you. We then wait a short time, known as a post-labeling delay time or inflow time, for the label bolus to arrive in our region of interest within the brain, and we acquire the image. And the basic idea with these techniques is that when we acquire the image, we want to have some way of distinguishing the amount of labeled water that are still in the vessels compared to that which have exchanged across the blood-brain barrier. And from this, we can therefore infer the permeability of the barrier to water. It's a, sorry, next animation, please, Daniel, sorry. And the next one, please. And the next one. Okay, so that's the idea. So having a contrast mechanism that allows you to separate the intra and extravascular component of the ASL signal in order to estimate how quickly the water molecules cross the barrier as a surrogate of water permeability. And just a few methodological characteristics of this technique that I thought would be worth mentioning initially is that this means, of course, the timescale of these measurements is across a few seconds, because this is an ASL technique, and this is very different to larger traces where the measurement of exchange takes place over several minutes. Secondly, this is tricky in white matter because of the low perfusion and high arterial transit times affecting the SNR. And finally, a difficult aspect of this method is the lack of accessible gold standard measurements for comparison. So if you use a larger tracer, you can get an analog of that, give it a fluorescent tag, inject it into an animal model, and then perform direct histological assessment as a validation. Such measures are much more tricky when it comes to water because of the tiny size of the molecule. The most commonly applied contrast mechanism is to combine ASL with diffusion, and this builds on the IVIM literature, where it was found that if you take diffusion measurements of the brain at different B values, and if you plot the signal intensity, you see there is an initial fast decaying component. This is attributed to the vascular bed that then gradually levels off. And when similar measures are done, focused now on the ASL signal, they find, unsurprisingly, that the perfusion fraction is greater, reflecting the fact that now we are isolating the signal from labeled blood water that's being delivered to the brain. And some nice data has shown that as we extend the post-labeling delay time, the perfusion fraction gradually decreases, reflecting, next animation please, the gradual delivery and exchange of labeled blood water from the vessels across the blood-brain barrier into the tissue. Next slide, please. Another contrast mechanism that's been exploited is differences in the T2 decay between the blood and the tissue, which is very much field strength dependent. And there have been studies that have applied this method. And if you look at the MR images, next animation, here you can see a short post-labeling delay. So these are ASL images. You can see that the signal is confined to the large arteries, and the rate of decay of the signal as we increase the echo time is fairly slow because of the high T2 of the arterial blood. And as we extend the post-labeling delay, this rate of transverse decay quickens, again, reflecting this transfer of labeled blood water into the tissue, as shown by the next plot. Okay, thank you. I thought at this stage it might be interesting to highlight a couple of notes of caution using these techniques that might present some interesting discussion points. I think there is an open question at the moment as to how accurately we can use these methods to separate the intravascular and extravascular contribution to the ASL signal. And so, for example, there was work by Sunji Kim, which combined magnetization transfer contrast with the ASL signal and compared with the diffusion, and they concluded that the arterial blood, rather than the capillary signal, is the main contributor to the pseudo-diffusion compartment that's observed in ASL. And given that intuitively you expect the majority of exchange to take place at the capillary bed owing to its huge surface area, this does raise some questions as to how accurately it can separate these two compartments. And there was another study by Timothy Duong and Sunji Kim where they used a perfluorocarbon intravascular contrast agent. So the black dots are the signal just from the vascular bed, not from the extravascular tissue. And they found that even at a B value of 1,000, they could only attenuate around half the signal from the vasculature. And this may be very different when it comes to water, but I think it is worth considering. And there may be a question also as to the directional dependence of the diffusion gradients. And this is something that we exploited in our own work where we applied diffusion crusher gradients to the ASL signal. And we found that the degree of vascular attenuation was very dependent on the orientation of the crusher gradients. And we could actually exploit this technique to generate maps of the microvascular flow patterns showing that we could capture this kind of characteristic radial patterns of the penetrating arterioles in the rat cortex. So it could be thus perhaps that the diffusion ASL technique underestimates the intravascular component of the ASL signal. Next slide, please. In regards to the T2 technique, it could be that the susceptibility effects extend beyond the vessel. And this means that perhaps the T2 ASL technique overestimates the intravascular contribution to the ASL signal. And also with the T2 technique, you're struggling slightly against sensitivity compared to the diffusion method, because the difference between the fast and slow decaying component is much subtler for the T2 method compared to the diffusion technique. And we did a rough comparison of the two methods, and definitely take this with a pinch of salt because it was only at 2B values, but we saw quite a big discrepancy in our estimated intravascular fractions using the method. So I think more work could be done to try to get to the bottom of this in the future. Next slide. And the next method I want to just touch upon is a very elegant technique called webcast. And this is trying to measure the extraction fraction. And as I mentioned, classical studies using radiolabeled water have shown that 90% of labeled water crosses the blood-brain barrier during a single pass. And in this quite simple technique, they just perform ASL and they look at the signal from the veins. And they also combine this with a phase contrast readout to reduce partial volume effects. And they've shown using this method that they have fantastic sensitivity to detect subtle changes in extraction fraction that occur with the caffeine challenge. And also they've shown good agreement with these techniques compared to measures of water permeability acquired using the contrast agent method. They've also recently published a work in JCBFM showing nice back translation of this method to the mouse brain. Finally, there's a paper on bioRxiv currently that describes a technique called vascular exchange imaging. This isn't an ASL-based method, but here they have a initial diffusion encoding block that kind of labels the signal from water in the vasculature determined by its diffusion characteristics. They then wait a mixing time to allow the water molecules to cross the blood-brain barrier, and they then try to determine which of these water molecules now have a different diffusion environment to try to assess the rate at which this occurs. So this is quite a promising technique that doesn't rely on ASL-based methods. Next slide, please. So I think I just wanted to highlight this study that was performed last year, because I think to understand what these different measurements are telling us about blood-brain barrier physiology, it's really nice when different techniques are applied in the same subjects. And this is a paper published by Danny Wang's group where he looked at an aging cohort and he compared DCE measures of gadolinium egress to blood-water permeability calculated using the diffusion ASL technique. And interestingly, they found that there was a correlation in these metrics only in selected regions. And so I think it'd be really interesting to try and understand whether this genuinely reflects differences in the physiology or whether there are some methodological reasons behind this. Next slide, thank you. Moving on now to application of these methods to aging, it looks like from the DCE literature that there is increased permeability to gadolinium in the aging brain. And this is shown very clearly from work by Alex Montagna and colleagues, where they see very convincing correlations between these K-trans value as people get older. In terms of the permeability of the blood-brain barrier to water with aging, the story is a little less clear. So we did a study in a mouse model of healthy aging using a T2 ASL technique and detected increased permeability of the blood-brain barrier to water. Similarly, the group from Manchester showed increased permeability of the blood-brain barrier to water in a rat model of aging, but recent work in the human brain using diffusion-weighted ASL showed the opposite relationship. And so hopefully, as more data emerges, we'll be able to understand this a little bit better. Next slide. In terms of application of these methods to age-related neurodegenerative diseases, DCE methods have consistently shown increased in blood-brain barrier permeability to gadolinium in a number of these conditions, including some examples are shown here, so including Alzheimer's disease, small vessel disease, and Parkinson's disease. And recently, some exciting breakthroughs within this field were made by the Zlokovic group, where, as well as showing differences in the rates of gadolinium egress in the hippocampus between MCI and control patients, the next animation, please, they also showed strong correlations between these measures of blood-brain barrier permeability in the hippocampus and CSF measures of this marker of parasite damage. So this was quite an exciting finding because it really does provide evidence that this non-invasive imaging techniques can provide genuine insight into pathological changes that are occurring at the blood-brain barrier related to parasites at the neurovascular unit. And similarly, well, not similarly, but they also showed that there was a correlation between these measures of blood-brain barrier permeability in the hippocampus and clinical scoring of cognitive impairment, independent of amyloid beta and tau status, again, suggesting that these measures could be very useful to provide real meaningful insight into the pathological processes that are driving clinical symptoms later in life, providing just a lot of encouragement that these measurements are very useful when studying dementias. Work by Ben Dickey and colleagues showed that there was a increased permeability to water in a rat model of Alzheimer's disease, and that this correlated with the degree of tight junction protein loss occludin within their model. And interestingly, they saw no differences between the wild type controls and the Alzheimer's model when looking at measures of DCE. Work from Danny Wang's group. This is now talking about the permeability of the blood-brain barrier to water in age-related neurodegenerative disease. They showed an increase in water permeability in patients with vascular risk factors, including diabetes and high cholesterol. And they also showed, next slide please, a correlation between measures of water permeability captured using the diffusion ASL technique and measures of CSF amyloid beta concentration in the CSF, which I think intuitively makes sense. If the barriers are less permeable, there's more opportunity for mixing of amyloid beta from the brain into these other fluid compartments. And finally, the paper which I mentioned, which is currently on bioRxiv, they applied the VEXI technique and they showed an increase in their metrics of blood-brain barrier permeability to water in MCI patients compared to normal controls, which provides evidence that this is a promising technique moving forward. So to conclude, these measurements of blood-brain barrier structure and function appear highly valuable for understanding the series of pathological events that drives age-related conditions. And I think the development of completely non-invasive techniques which don't require contrast agents would be very useful to enable more practical application of these methods in a wider variety of patient cohorts. I think greater technique validation and characterization is required to understand some of the discrepancies that I've shown in this talk. And a way to help with this is by comparing different techniques in the same cohort of patients, as have been done in some example studies shown here. I think preclinical application to mouse models with specific changes at the blood-brain barrier can be useful to understand the physiological basis of changes in blood-brain barrier permeability that we see. And finally, just being able to try to better account for possible methodological and analytical reasons why there are differences by, for example, having the same person analyze different data sets and making data publicly available would be really useful. Finally, I'd like to thank Dengrong very much for advancing my slides so well. I think we had quite a good connection there, so thank you so much. And I just finished by just thanking David Thomas, UCL, and Laura Parks, Benjamin Dickey, and Yolanda Heaney for their input in preparing this talk. And just want to highlight two fantastic reviews that were really useful when I was putting this together by the Manchester group. And thank you all for listening.